Hello and welcome back again. This is Ms. Bushra with you, and I'm back again with another lesson. Today is the second part of the specific immunity, which is section 2, chapter 3. Let's not forget. We've learned about the organs of the immune system, which is part of, the specific immunity. The lymphocytes that are all made in the bone marrow but then develop in different areas where they get their names. But before we start how they are working, we need to know what is the trigger that starts all that. Why specific immunity is not always activated. Okay? What is the specialty? What is the mechanism that make it so specific and make it so special? So the first thing, look at the title. It says recognizing a pathogen. The whole system depends on this. If you cannot recognize pathogen, there will not be any reaction starting from the specific immunity cells, lymphocytes. So lymphocytes are part of specific defenses because they are able to recognize foreign invaders with their ID. Lymphocytes can decide whether this is a flu, an HIV, measles, whatever it is, they can recognize it. Okay, let's see. This is like probably not new for you, but let's hear it again. What is an antigen? Any substance that the immune system does not recognize as part of your body and it's not similar to us, it's considered antigen. Antigen could be a microorganism, could be a piece of this microorganism, okay? Could be the only protein, okay, the proteins on the surface of these microorganisms. Could be a tissue from somebody else's body that's not yours. Like blood, we've studied about that. It's considered antigen. Like a transplant, for example, kidney. This is one of the main reasons why transplants are hard. You cannot just get it from anybody because it triggers your immunity. Since each one of these things have one thing in common, is that the surface of their cell have this projections made of proteins, glycoproteins, a lot of stuff. They're considered antigens. As long as they're covered with these, even an entire organ is considered an antigen. Okay, so there it is. There's everything. Even pollen, which is part of plant, is considered as an antigen because you did not create it. Toxics and venoms, these are not part of your body. That's why then some people, when they have like developed immunity against toxins, they say we did that, we exposed ourselves. It's because they make sure that they just get enough of it, that they don't get the harmful effects, but their immunity creates antibody for it. This is why we consider them antigens too. Remember, transplant tissues, organs, transfused blood is one of them too. So when do a specific defense begin responding? With recognizing. So the key for responding, the key for activating this system is recognizing what antigen came to our body. Okay? How do we recognize? They need to bind. It means when antibody bind to antigen, it's considered a reaction and it's considered recognizing. Okay? Binding, recognizing, it is a reaction. Don't forget about this. Okay? So, a reaction of the body against an antigen is known as immune response. So, the whole process of having an antigen and then an antibody finds it and binds to it, or like anything that's a receptor in the body finds it, it is considered the first step. Then afterwards, antibodies are created for this antigen. This whole stuff, at the end, what do we call it? There was an immune response against it. When do specific defenses begin to respond? Remember, before I start anything, our immunity, either it is the white blood cells or even like the Y-shaped protein, the antigen. What do they have? They have one thing in common in both of them. is that both of them have receptors. What is this? Receptor. Receptors have complementary shapes for antigens. Let's say that a virus comes to our body that has an antigen like this on its surface. Another one also at the same time comes to our body. Let's change the color. Has an antigen like that. Okay? You can clearly tell which one is the complementary one. See, these receptors, don't forget, this is receptor. This is antigen. This is antigen. AG stands for antigen for me. Okay? These two don't match. They don't match. So no reaction, no binding, no immune response. However, even with this one, because they have the same shape, this is complementary for both that and for this one as well. That means there will be a reaction because they combine, they're complementary in shape. That is considered a recognizing. Without this binding between them, there's no recognizing. Okay? How do they bind? Remember, the receptor looked like this. The antigen, let me change the color, looks like that. See? Complement shape. They're sitting, okay? They are like able to bind like that together. Very good. So the beginning of every immune response is recognizing. How do we recognize? Binding of complementary shape, antigen and receptors. Let's not forget about that. This is considered a reaction, and this is considered the beginning of the whole process. There you go. When do a specific defense begin to respond? When a lymphocyte has a receptor. We studied what is receptor. Let me minimize it. Okay, has a receptor protein all over its surface of the cell membrane. We know that. These receptors can recognize and bind to an antigen in a three-dimensional way. What I drew was like a 2D. It's a diagram. But in reality, they're like a 3D shape. They fit into each other. They're perfectly fit for each other. Very complicated as well. For instance, the surface of a bacterial cell covered with many different kinds. Please highlight this. Antigens could be many and could be multiple on the surface of a pathogen. Okay, or any antigen. There are like so many of them. But a lymphocyte carry only one type of receptor. Okay, so this is possible for another antigen to be here. Okay, another shape here as well. But this is a pathogen. But every lymphocyte carry only one type of receptor protein. This is very important to know. Okay, very important. This is what makes it specific. If I asked you what makes lymphocytes so specific, they carry only one type of receptors, and their receptors are matching only one type of pathogen. In case of NK macrophages, they have like no specific shapes. They could recognize any of the other antigens as well. But here they can recognize only one antigen, and very specifically, their shape is perfectly complementary. You see, in this case, we have three examples here. Each one of them have their own type of a receptor, but all the cells is covered with one type. Let's not forget about this. Then, these two pathogens were introduced to this group of lymphocyte. You see that two of them started reacting because their receptors were complementary shape to the pathogens. But remember, they can only bind to one type of pathogen. This cannot go there. They will not bind to each other, okay? And this cannot bind to this one. This one doesn't have a receptor that match either one of these. It is super different than both of them. It has another pathogen for it. So there's no binding. So if I say why, this was in exam 2025 by the way, why this one did not bind? You're going to say because the receptors on the surface of that lymphocyte did not match the antigens of the pathogen. That's all. You can write it as a note. This is the core of the immune response. If you understood, you're good. Let's revise it one more time. What is the first step of any immune response? Is recognizing the antigen. Who's recognizing it? Lymphocyte recognize it, okay? Lymphocytes have receptors on them that is complementary in shape to a certain antigen. How many types? Only one type of receptor is on the surface of lymphocyte. So for a lymphocyte receptor to be able to recognize an antigen, they need to be complementary in shape. Once they're complementary in shape, they can bind to each other, and this is the onset of the whole immune response. What if this lymphocyte does not have a complementary shape receptor for that antigen? There won't be any reaction, there won't be any response. But that doesn't mean we are going to leave the antigen. There will be another lymphocyte coming, probably this one is going to match it, because there are millions and millions of lymphocytes, but each one of them carries its own specific shape. So your body can find like a lot of species of different organisms. This is why it says how the body defends itself. There's enormous army of pathogens outside. It's because your body have, like we said, millions of kinds of lymphocyte. Each can find one type. Another thing, so when is the body infected with a specific pathogen, are all lymphocytes able to answer? No, they cannot do that. There, like I said in my story, there must be one with a complementary shape to that specific antigen, and this is what it says. For example, it says a cold virus enters the body. Lymphocytes that have receptors matching the antigen of the cold virus will respond and recognize it. But lymphocytes with a receptor that is matching a flu virus could not find out whether this is a cold virus. It doesn't care. It's unbothered with anything else. They only focus on one type, and that's it. Immune response. Since we're saying this is the onset of immune response, this is the beginning, let's see what is immune response to begin with. An immune response is made of two things. It's cell-mediated and humoral. Cell-mediated is where a cell of our immune system kills a cell in our body that is infected with a pathogen. Remember, immune response kills two things. Either infected cells or we kill the pathogen directly. In this one, we kill the infected cell by an immune cell. Here, it's where antibody produced. Here is antibody produced. And since antibody is in the plasma, we call it humoral. Humoral means the fluid one, okay? The fluid one, it means the antibody is in it and it can kill. Antibodies are actually helping the process. We're going to learn later on. It's a bit complicated. So let's see. One thing, these are not two stages following each other at all. They're actually two processes happening at the same time. They get activated by the same thing. And the key factor for both of them is helper T cells. Without helper T cells, this process is not going to get started. So how do we tell helper T cells to start a process? Again, with recognizing. So let's see how is that possible. The first thing, I'm going to explain it on the picture. I would rather you use the picture too, and you use the descriptions just for your own information. The cell-mediated response is written here, but actually the three steps in the beginning is shared between both cell-mediated and the humoral. Cell-mediated start from 1, 2, 3, and the two specific parts are step 4, step 5. However, in the humoral, step 1, 2, 3 to activate it, and then step 6 until 9 are part of this humoral response. So you can tell that this is a shared step between both sides. Okay, check this out. We start with this. Do you see this cloud-like structure? It's a macrophage. Okay, This is a macrophage. Naturally, we learned that macrophage starts engulfing microorganism. This is our microorganism, which is a virus. Do you see these green stuffs on the virus? This is what we call antigen. When you say viral antigen, means an antigen for a virus, okay, for that specific virus. But look at this. We did not only label it here. Why do we label the macrophage for viral antigen? That's because when macrophage eat the pathogen, or especially in this case the virus, it takes off these antigens and put it on its own cell membrane. See, this is the cell membrane of the macrophage, but you can see all over it there is antigens of the virus. This is not what macrophage made it itself. It took it from the virus and put it there. Why? It's very weird, right? But we call this action displaying. That's why in some books they call macrophage the antigen presenting cell. It means like it presents antigens of its enemy on it is, surface membrane. It's just like if when they are, let's say an army is taking over a place and colonize it, but then they do what? Let's say they put a flag on top of it, okay? This is marking, and this is what we're doing. Displaying is just like putting flags that this macrophage has already take over a pathogen and we need to recognize it. Why this is helpful? Because it's easier to find a macrophage than looking for a virus. Who's going to come here? With chemical reaction, it will attract T helper cells. The T helper cells, what do? They have receptors. These are receptors that are complementary to the shape of this antigen. That's why there is what? This is binding. Binding means there was a complementary shape and the recognition is done. Now we know we have what type of virus is here. Let's say this is HIV. Now this lymphocyte is special for HIV. Found it. But what is the name of this one? It's called T or helper T cell. T helper cells. It's the same thing. The T helper cells binding with the antigen on the surface of the macrophage tells the macrophage there is actually a lymphocyte for it. So it starts releasing a certain type of chemical called interleukin-1. Interleukin-1 does two things here. It is going to make sure that there's more T lymphocytes to start the process. So it makes these T lymphocytes start doing what? Multiplying, so that their numbers increase. And also activates them, because they were not activated, activates them with this interleukin in order for them to release another interleukin called interleukin-2. So that's two functions you can write. First, upon the binding, there must be binding before the macrophage release this. Binding, then release of interleukin-1. Interleukin-1 makes sure that these number increase and they release interleukin-2. We have it in the table in the next pages, by the way. Okay. Why interleukin-2 is so important? Because the interleukin-2 starts both humoral response and the cell-mediated response. Without interleukin-2, we will not have that. We had in the third step interleukin-2. That's why we're going and study the cell-mediated. Here is cell-mediated. Now, the interleukin-2 is going to affect a certain type of cell called cytotoxic T cell. This is a type of T cell, okay, but a bit more specialized. You could tell there's like some receptors here. The cytotoxic T cell could go and look for infected cells, not pathogens themselves. Cells of our own body that pathogen went inside it, we call it infected cell. It's going to find it and then rupture its cell membrane and kills it. But how did it know that this exact cell is infected when another one is not affected? Do you see the same viral antigen? So these cells, once a pathogen goes inside them, they know, okay, we're infected, we're gonna die. So I might as well tell the immune system to come and kill me. That's the process. So this white normal cell that got infected put the virus's antigen on its own surface, just like how macrophage did. And what did it do? It made it easier for cytotoxic T cells to destroy them and kill them. How does it kill them? By rupturing and piercing its membrane. This looks like how the natural killer cells also doing the same thing, and also complement system proteins. They're all putting a hole in the cell membrane of the infected cells and kill them. Okay? This was cell-mediated response, the 3 steps and these 2 more steps. However, the 3 steps where we had interleukin, if you notice, the interleukin further activate another cell. What is this cell? It's the B cell. B cells get activated by interleukin 2 and starts multiplying. They start increasing. However, when they multiply, they don't become one thing. They become 2 types of cells. One of them, which is the fewer number, that's why it's called sum, it's called memory cell, which is very important. These memory cells are not going to fight and not going to participate. They're keeping information of this process. They keep information about the antigen and what receptors they needed, and they're just going to stay there. In the first time when you get a disease, these memory cells are created and hold all the information possible. When the second time comes around, they are the ones saying, oh, we already have the information, let's do this. That's why the immune response in this later on, other than the first time, is very quick, because you have memory cell then, okay? They hold memory of what happened before, and you don't need to worry about if this is a new virus or not, because they're the one taking care of it. There you go. B cells here, another batch of them becomes what? Most of it. That's the higher number. Okay? Plasma cells. Plasma cells are the cells that are becoming, okay, the factories of antibody. Okay? So if I say what type of cell is creating antibody, you're going to say plasma cell. If the option did not have plasma cell, then B cell. Why B cells? B lymphocytes? Because B cells are the general name for both memory and the plasma. Plasma is just a variation of the B cell, okay? So this is extra. Antibodies were produced, and you can tell the antibody could either, on both, because we have 2 receptors here, they have a white shape, okay? They could either attach to 2 different cells or same cell here. What do they do when they attach? They neutralize it and mark them. What do? Neutralize and mark them. So antibodies, they mark them for destruction. What's that supposed to mean? It means they don't do it by themselves. They're just becoming a flag that the macrophage later on sees anything with these attached to it, that means it's for destruction. Okay? So a macrophage comes and eats them all as a batch. That's it. We're going to, talk about the others in details. Let's see, this is a space for your notes. There is something. Remember when we talked about interleukin and interleukin 1 and 2? What are they? They are a group of proteins and chemicals that called cytokines. These are proteins of defense mechanism, but their job is not fighting directly, but rather they're affecting the behavior of immune cells. They change it. One of them is not activated, it activates it. One of them is not multiplying, make it multiply, okay? There's no cytotoxic T cells or B cells working and producing antibody and fighting. It's activating them. This is changing in behavior. But you need to know which one is released by which cell. Interleukin 1 is released by macrophage, while interleukin 2 is released by helper T cells. Another thing, macrophage will not release them unless there is a pathogen that was engulfed and then the binding of T helper cell to the antigen on its surface. Without these 2 following each other, you won't have interleukin 1. Now, interleukin 1, it activates T helper cells for what? First, releasing interleukin 2. Second, increasing in number. And third, it's causing fever too. Okay, remember when we said there's a chemical causing fever, macrophage release it? There you go. Now, Interleukin 2, it activates production of more helper T cells, cytotoxic T cells, and stimulates B cells. So all three of them. That means a shared feature between these two is that both of them make sure that there will be more number of T helper cells. It's a crucial and key factor of the whole system. We're going to study in HIV how HIV is messing up with the helper T cell. That's why the whole immune response will stop. Okay, it will stop. There is all the descriptions, definitions of each one of these T cells, the T's and the B's, all of them. There's this is lymphocyte, what is releasing it, what factors or like what part do they have in each system. It's very important. Remember, cytotoxic T cell can kill infected cells of our body and also cancer. Remember when we say specific immunity can control cancer? It's because we have cytotoxic T cells, okay? It kills cancer cells and infected cells, also parasites, any foreign tissue. So one of the reasons why tissue transplant, kidney transplant, other organ transplant might fail is because of cytotoxic T cell. There is one that we did not mention it in the pictures. It's called suppressor T cell. It's another type of T cell. This suppressor T cell, okay, check it out. It is to stop and shut down this process once there is no more pathogen, okay? Because this process might go on if it triggered, it's better be controlled. Remember, B cells is becoming these two, okay? They're not separated. This is just the general name. It divides and becomes these two. What does the plasma do? It is highly specialized and they create what? There you go, antibodies. Up to 30,000 antibodies per second. That is amazing and a huge number. Remember, memory cells, they are lymphocytes, B cells, of course. They are created and generated during first infection, but only starts participating in the process of immune response from the second infection and the later ones as well. Tamam? Never participating in the first time. Remember, what was antibody? It's the Y-shaped protein. And it has two receptors at each end. These are called identical arms. And these, each one of them can bind to an antigen. It's possible for the antigen to be on the surface of same cell or different cells. We don't have a problem with that. Okay? What do? When they bind and attach to them, they are just blocking their activities. That's called neutralizing. Okay? So write down that the antibody's function is to neutralizing, which means, write down as well, does not let the pathogen multiply and at the same time does not let them infect any other cells. Number 2, they are also marking them for macrophages and complement system to come and destroy them. And it's written here. Okay, that's why we say that complement system is part both non-specific, okay, when they encounter an antigen by themselves, or part of specific response only if there is antigen-antibody reaction. Okay, then it participating in the specific immunity. You can clearly see there are like some differences between humoral response and cell-mediated response. Okay, so the cell-mediated response, it is making sure that we're controlling the numbers of the infected cells here. The actions are done by cytotoxic T cells, and it kills any cells that invaded, got invaded by the pathogen. That means infected cells. The cells are participating are T cells, but here we have B cells mainly. T helper cell, a shared feature between them, and it kills the pathogen with antibody work. Okay, here we don't have antibody. We have antibody. All right. Here T helper cells participating. Yes, T helper cell as well. The cytotoxic T cell and suppressor. Okay, the B cells, memory and plasma. Okay, this is how you differentiate between them. There you go. Let's start here. When does the immune response come to an end? Once there is no more pathogen, so your body is very smart, does never, okay, it's never going to produce more than its own need of anything, and it's never going to start a reaction and just go on like that. It will stop at one point if the trigger is not there. What is the trigger? There's no more antigens or infection in the body. So how cytotoxic T cells and natural killer cells are similar? Both white blood cells, okay, and both kill cancer cells and infected cells, and both of them are doing it by piercing the cell membrane of infected cell or cancer cell and make the water go inside, the content coming outside. This way they kill it. Okay. As for cytotoxic T cells and natural killer cell differences, one of them is non-specific, the other one is specific. Which one was non-specific? The NK. Which one is specific? The cytotoxic T cell. Another thing, this one is killing any infected cells, any. That's for the natural killer cells, any. Not specific about it, does not matter, but the cytotoxic T cells need to identify and their receptors must match the ones on the surface of the infected cell that got it from the antigens. And here we're going to end our lesson. I hope you understood everything. If you have any questions, please comment below. If you need anything, just comment below again. And have a nice day. See you next time.